Okay. Hi, everybody. It's nice to be back on the user stage again. It feels like a school trip with many friends that I already met in Aalborg, and it's good to see them, many of familiar faces. So, as Laura said, I'm quite honored that I'm able to present here with some of the results of my thesis, and it's going to be about, say, the development of applications that go for doing interactive and reproducible research in RNA sequencing applications. So, I'll start broad with science, data, and analysis. And in the latest years, there's going to be there has been a lot, explosion of complex data that have been generated in any scientific discipline. And I can ask you to raise the hands of how many of you have more data understanding than data generation. Probably not that many. So lots of data are just standing there and ready to be analyzed. And the computational analysis is become a standard component in basically any scientific workflow, to the point that you can actually call biology also quantitative science. When you do data analysis, there's not so many things around that actually can grant you some robust and accessible analysis at the same point. And this is a key issue in bioinformatics. When you have to deploy or adapt or develop tools, so you have to take into account that each of them has a set of capabilities and limitations, and these might depend on the application, on the audience that are supposed to use. And as a wish list, we are here and we want things to be comprehensive, interactive, and reproducible. For the comprehensiveness, we would like to have end-to-end workflows, and since we develop or like our packages, we are on the good track for that. And I will touch upon interactivity and reproducibility just in a second. If we sit here as analysts, so we are just in the process of going from raw data to knowledge, which sometimes it's oversimplified, and a lot of people think that batch processing in a blind way is the best way to do it, but it's not, and a lot of exploration efforts go actually quite underestimated. So interactivity is actually essential from going to raw data to knowledge, because it really does empower the domain expert so that he actually can get better insights on his own data. And it, of course, makes it simple. So you can go from complex data to efficient visualization, ideally, in a few steps. So it's a paradigm which is easy to adopt. As for the reproducibility, I'll stick to the technical definition of it so that you can actually reperform the same analysis with the same code on the same data. And if you want to try to move along these two axes, of course you start here, and then you want your results to be replicable if you use different data for doing that. If you use different code for reproducing the same results, then it might be that it's robust. And if you have both of the things, then your results are generalizable. But I'll stick to the technical part of it. And this is probably the best tool that we have to actually fight back the reproducibility crisis on this technical level. And this transparency that it's granted by this, it's essential because you can do independent external verification, and on top of it, it's the only thing that actually grants you the possibility to grab a method, have new data sets, and truly stand on the shoulder of giants so that you can really progress in the scientific advancement. For those of you who are not familiar with RNA sequencing, I'll start with the ABC and then move on to EDA and DE. So some of these acronyms they will be known for you, but I will explain all of them. So the ABC, you go from you go from DNA, which is transcribed into RNA, and this is just the exonic parts are then spliced together and compose this protein, which are in the end the effectors of the plethora of functions that happen in every single cell in your body. And from the analytical point of view, this turns down to a lot of text, which is then structured in reads, which get aligned to your genome. So you just need to find out where they originated from, and it ends up basically having some kind of profiles like this, where you can actually quantify how many of these reads are actually mapping to these gene of interest of yours that you can exploit from the annotation. And long story short, I glossed on the details, but in the end you have a matrix of counts, which is what you process afterwards. I've applied this concept to the platelet transcriptomics field, which is per se actually quite interesting because it sounds like a contradiction. Platelets have no nuclei, but still they carry a super vast repertoire of transcripts, so from mRNA to microRNA, circular RNA, and so many. And in the lab, which is led by Professor Wolfram Ruf at the CTH, they're working on the broad concept of cancer-associated immunothrombosis. So there are different scenarios, for example, the alteration of thrombin signaling and what happens, for example, if blood clot more on that. And these can crosstalk along with how tumor develop. So for them and with them, I did a lot of explorative data analysis and differential expression analysis. My aim is to create web-based applications where interactivity and reproducibility really do work together. It sounds like trivial, but it's probably not. So when I was looking around and looking for, say, potential existing solution, I couldn't find that many. And the idea is that this is the main component to get efficient analysis, good data visualization, and integrate data efficiently. You can also, of course, allow for transparent and external verification of whatever you did, all the steps and all the applied methods that you applied. And it's quite already a good direction into writing the material and methods section of your paper, which was also an advantage. Sorry for the time. So right now it can make you hungry for this. But so this is also my aim. This is your analysis. It would be nice, beautiful, and of course on top of it, it would be transparent. So you see all the layers. And to the question where do you want to take the cake or bake the cake? Of course, biologists would probably answer you, I want to take the cake, of course. But with this interactivity and reproducibility, I also try to address the fact that it can be a pedagogical tool. So people can actually learn something. Because, in a second, my recipe for this, I do combine interactive, so literature and reactive programming, and I exploit the software ecosystem of Bioconductor, and I also address the problem of deployment in this. So I use knitr and Markdown for literal programming, Shiny for the interactive part, and Bioconductor for this. So imagine yourself sitting in front of the data, of your RNA-seq data. The analyst will be fine with R and Bioconductor packages to generate the analysis, and then we just create the report. But we needed these extra layers of Shiny and R Markdown to make it more interactive. I'll spoil what's going on under the hood. So the solution is based on the following things so that you can also have some more hints to follow. So I have an embedded text editor in the Shiny app, which leverages on the Shiny Ace package. I fight the white sheet syndrome by providing a template report alongside with a package which is editable if needed in the Shiny Ace editor. And I also include a preview of the report live in the app. R Markdown just grabs the reactive values live in the Shiny app, and when it's screening and rendering, so it just displays them, adds it in the latest state. And before actually our studio implemented a clever bookmarking system, I thought of saving and sharing the binary objects or the global environment, or say whatever things that you had, I wanted them to be saved and shared with other collaboration partners and or colleagues and stuff like that. So one more thing that I wanted to address is I really wanted to make it usable. So I borrowed the idea from the Galaxy framework, which has moved from YouTube videos for guiding the users to interactive tools based on JavaScript libraries. I will show you just a couple of them in a while. The real motivation basically was to get a little lazy and or outsource back the analysis to the biologists, and that's why the reason was to please data, tell me more, and tell me more whether the data has just a car or just a real heart in that. So the two packages that I will be talking today one of them is PCA Explorer, and it's available in Bioconductor. So principal components analysis, for those of you who wouldn't know about it, it's probably the most widely used dimension reduction. Sorry for the HNMF, it just came out. But say you project on a 2- or 3-dimensional plane your data so that in such a way that they spread along the direction that explain most of the variability on your data. And I felt that it was a general downweighting on how important data exploration would be. And I say again, it's for discovering association of patterns in high-dimensional settings. It's essential. As a wish list, since I was developing that from the scratch, I wanted usability, reproducibility, and full integration with Bioconductor. And surprisingly, I got some bug reports and even better pull requests. So I'm very fine in gathering a critical mass of people that want new features, and I'd be happy to implement them for and with you, for example. And moving on to the next one, which is called IDEAL, and I was surprised that no one took the acronym for that, which stands for Interactive Differential Expression Analysis, LOL probably. And differential expression, for those of you who don't use it, it's the essential step to identify what are the molecular drivers of the observed phenotype. And biologists still do use Excel for that, but they shouldn't. And I leveraged the ESEC2 method for that. A list of genes is not enough, and that's why I want to take the information layer to pathways, so some kind of extra function interpretation. And again, I was aiming to create a platform that allows you for reproducible analysis, so full report and state saving. And of course, the self-paced learning, which is triggered by these interactive tools. And thanks you for Carl Ganz, if he's listening or if he's here, for implementing the beautiful R-intro.js package, which I use for that. So to use it, you just need two things, your count matrix and the experimental design information. And then you just launch the package, so it's 2 lines to install, 2 lines to launch, and there is some kind of layer of documentation. And if you want, instead of using, say, cryptical gene annotation, gene identifiers, you can use gene symbols, and I can just play that for you and see if it really looks like this. So I didn't want to challenge Murphy's laws too much by doing a live demo, so please watch it with me. So I have a data set where I have 2 different conditions. So I have a wild-type mouse and a mouse where a tumor is induced. So altogether it's 13 samples, and I want to check, for example, how the counts table that I submitted looks like. You can go to the normalized values, and it's just an interactive table for that. You can have an overview of your data. You can select whatever you want to color by, so your condition in this case. And you noted that there is this strange sample that seems to behave quite weirdly. So you wanted to check a little more. Since it's platelets, there is quite some contamination of leukocyte, which is possible. So we check, for example, for the integrin alpha M gene. And indeed, we see that there is this very strange sample, which is probably just contaminated. So yeah. We would like to check more on the samples view, and indeed, we confirm it also on the PCA level. And the interesting thing is any of these plots and any of these tables, you can just click a button and then say obtain a vectorized graphics or version for that. You click on those, and then you can zoom in. And then you can, for example, see what happens if we just remove that samples, and you see that indeed the samples still do not cluster perfectly, but it's just getting better. So your first principal component now corresponds with your biological condition. It's robust to the number of genes that you include in the PCA Explorer, in the PCA exploration. So you move on to differential expression analysis. And for this, there is IDEAL, which is structured in a slightly different way. So there are these boxes up there, that basically tell you what data you already have and how you should create it. And I'll show you how to use it. So we upload the data for that case, where already 1 sample was separated. The design, you can select it and create the ESEC dataset object for that. You can retrieve on the fly the annotation for that. It's a mouse dataset. You click on that, and then you see that the annotation turns green because it's now there. You click on the Run the ESEC button, and magic, flash forward, and you just generate the results. And I can show you now how it looks like when you use this intro.js library for doing that. So you can just say condition, okay, and I want to see how to use it. So indeed, basically, you just move along all the elements. It displays you what you have to do, and you are kind of forced to do what you're expected to do. So there's no way out. You actually have to reuse it in the right way. And this is quite cool because it's, apart from, say, pop-up that actually tell you what to do, this is even better because it forces the user to try that out. And so let's say tumor versus wild type, and you kind of have additional option for generating the results, but then you get a table, and it's populated also in the DE genes box, and you see you can have an interactive table with, you can sort it by logfold change, you can click on the gene names, and it will put you to the PubMed and Ensemble annotation on their database. You can have an MA plot as a summary overview of this. You can zoom on it, and if you provide the annotation, then you also see the gene names, which should visualize over there. So you might have your genes of interest, and you can just get some information of tropomyosin 2 and or thrombospondin 1, and you have some information over here. If you fancy volcano plots more than MA plot, it's be my guest, and otherwise you get heat maps in a static and interactive way. And the gene finder, which I already showed you from the PC Explorer, this is a little expanded because you can have more genes that you may be interested in at once, so you can either select them there live, or otherwise you can provide a text file, and then you get, say, box plot for all of them, and all exportable, and on top of it, a summary MA plot with all the genes which are annotated. So this is a typical example. You want all the chemokines to be included, you can just do it right away. Interesting, if we're going to DE genes to regulated functions, you take the up and down regulated genes, then you use the TopGO method for that, which is quite nice because it really gives you some targeted GO terms which are meaningful. And again, with a flash forward on that, I'll skip to the report editor part of it, and I show you how it looks like. So there is a text editor, which should appear now, and you can just, it looks like RStudio. There is also auto-completion of the code. That's the template that I provide alongside with the package. And if you click on the report, on the update report, you can just show it. So right now, these functions are now computed, and you can get a table, again, interactive. If you click on the table, you can get, for free, a heat map of the signature for that genes in your data right away. So this is a typical use case that, say, normally otherwise would take me quite some time, and I outsource it back to the data generators, which is good. So the report looks like this once you generate it. So you have a preview in the app, and it just looks like, so whatever selection that we did, it's just reported over there. So there is, the whole analysis is still stored. And you can just scroll it and check whatever, everything you want is in there. Otherwise, you can just go back to the editor, and you can edit and can regenerate it. And again, everything that was there is now in the preview of the report. Additionally, you can click on Generate and Save to actually export and download the HTML, or you can click on this cog, and you can, for example, save your state either as a binary data or to the environment. So that would be it. So you can just then see how it looks like once you generate the report. It's nothing else but the thing that you had in the app, but you have an HTML that you can just save, share, store, whatever it is. So I go back to this, and I hope that I was able to convince you that interactivity and reproducibility can actually be combined together in an efficient way. And this makes the whole analysis accessible and reproducible, and you can extend this approach to many other applications. The report is there, and state saving is also for granted. And I wanted to make a focus on easy to use and easy to learn software. There's a paper from a colleague of mine which I also cooperated with, where we defined this criteria for that. And you can do a lot of data forensics and advanced differential expression and downstream analysis with your data. I would like to introduce new dimension reduction techniques and extend this to single-cell RNA sequencing and or to move from gene to transcript level information. If you have better ideas on how to enhance the reproducibility framework, please come and talk to me. I'm happy to improve my package. And for those of you fans of the series, so the ideal way of doing RNA-seq analysis is not coming, but it's already there because the packages are available in Bioconductor. You can try them out. There is a Shiny server which runs all of this and it's publicly facing, so you don't even have to install that. And to practice what I preach in terms of openness, everything is available at GitHub. Sorry for running out of time. Just thank all the nice and clever person that I work with, and I thank you for your attention, and happy to take questions probably in the break.